Point, you should have looked at the assignment that we're going to be working on right now. We're going to be modeling the process of meiosis, which is how gametes are made. Gametes, they are haploid cells, but they do start from a diploid cell. As we go through this investigation, we're going to be looking at the process of meiosis without something called crossing over, and then we're going to go through the process again and include crossing over. And essentially what we're trying to look to see is at the end of meiosis, when the gametes are made, what things during the process of meiosis contributed to all gametes being genetically unique or not the same as one another. Your group received a bag that had this card in it. This is kind of your key. It explains everything that's in your bag and what it represents. So for example here, the yellow pipe cleaners in this bag, you might not have yellow, but the yellow pipe cleaners in this bag represent chromosomes from mom, and the blue ones represent chromosomes from dad. And if you look at these, the blue and the yellow, they are exactly the same, same length. So that's going to indicate that these chromosomes, they are homologous. They are the same. There's also 2 pink and 2 red ones in this bag. Again, the pink would be chromosomes from mom and the red chromosomes from dad. Those 2 are the same length. Therefore, they are homologous. Each homolog consists of 2 sister chromatids. They typically would have an X shape. But we have to put some genes on there, and that's what these beads represent. They represent different genes. Here's an explanation as to what the colors represent. And on this one, I'm going to put a trait of eye color and a trait size of ears. And it looks like there's 2 variations of eye color. There's green eyes and there's brown eyes. Green eyes are represented by the orange beads. So here's my orange bead. I'm going to put those on this chromatid here, and I'm going to put size of ears. I'm going to go with large ears, that variation large, which is the green. So I got my green one here. So I have my first chromatid set up, and I want to point this out. I didn't do this one yet because, well, in G1, we just have 1 copy of this chromosome. And we'll have 1 copy of the blue one too. Didn't set that one up yet. But in the S phase, this chromosome, it is duplicated. So I'll take my second sister chromatid, I'll put on my orange one and my green one, and then these 2 sister chromatids, they are identical, and they would kind of take on that characteristic X shape like so. And again, my orange beads, that gene is for orange, the trait is eye color, and the orange bead will create green eyes. The green beads, the trait that one codes for is size of ears. Variation, these will create large ears. Let's see what the blue ones will kind of be. And since I used orange here, which is eye color, I got to use the other eye color one, which is the yellow bead. So I got to use my yellow beads on the orange one. And since I did size of ears here, I got to use the second size of ears. Small is the variation, and those are the blue beads. I'll put my blue one down here. And again, this chromosome, it is duplicated in the S phase, so here's the process of replication, green and my blue, and then that would kind of take on that X shape. Repeat for my other 2 chromosomes, and then we'll be ready to go. I'm going to pause the video, get that set up, and we'll resume when you see what it looks like. All right, so I have my chromosomes all set up. On my small ones, it looks like I have my pink beads. So this would be a trait of hair texture. Variation is going to be straight. And that means my white ones will be for hair texture as well, but not straight hair. The variation is curly. My purple beads are for trait of size of the nose, variation small. My black beads, size of nose, variation large. We're almost set for our first photograph, but we have to do a few things here. First, we're going to show prophase one. In the process meiosis, we go through the same phases as mitosis, but we do it twice. So there's going to be prophase I, metaphase I, anaphase I, telophase I, and then we're back to prophase II, metaphase II, anaphase II, and telophase II. A few things that I want to kind of label in here is I do want to mention that these two chromosomes, they are homologous. These two chromosomes, those are homologous. A little bit of other information on here since we're just getting started. The orange beads, those are for green eyes. I just kind of combined the variation along with the trait, and I see orange beads, green variation, eye color, so green eyes is how I got that. On the homologous partner, the yellow one would be brown eyes, so those are brown eyes. The green bead, let's see, we got large ears. My blue one is probably going to be, and it is, small ears. My pink one, straight hair, which means that my white one is going to be curly hair. And last trait, my black bead, looks like that is a large nose. And then my purple one is going to be a small nose. We're not going to be labeling these in every single one. We're going to just kind of do in the first one, and then maybe at the end just we can kind of see how things separate here. But again, this is prophase I, and this would be the first picture that you would take now that you have your board set up like this. Once you have your picture done, we are going to proceed into metaphase I, see how these chromosomes change. So you might want to take your picture, pause this video while you do so, and then I'm going to erase my board. Let's see what happens in metaphase I. And again, we are doing metaphase I. This one is going to be without crossing over, because we're going to see the effects of when the chromosomes do cross over versus not, and essentially find out the significance of crossing over. So metaphase I, just like in mitosis, the chromosomes line up in the middle of the cell. However, let's check out what this would look like in mitosis. It would look something like this. The chromosomes line up in the middle of the cell. That would be mitosis. And again, in mitosis, we want to get one of these sister chromatids into each cell, so we're going to separate the sister chromatids. That's not what meiosis is all about. In meiosis, we're trying to create gametes for reproduction. And we know that we need to get one of these homologous chromosomes into each cell. We have to make sure that mom and dad, only one of those two chromosomes goes to a child. We talked about Down syndrome. That would be the case of they get the chromosome from both mom and dad. So the homologous chromosomes, they line up in the middle of the cell during metaphase I. And this will be metaphase I without crossing over. Again, we have our homologous chromosomes in the middle. And then we have these two homologous chromosomes as well. I'm going to just write the word homologous for simplicity moving on. Again, here's metaphase I. Take your picture, and I'm going to clear my board. We'll look at anaphase I. Anaphase I. Again, let's check out what happens in anaphase I. Again, this is anaphase I with no crossing over. And in anaphase I, this homologous chromosome gets separated from its partner. They go to opposite sides of the cell, like so. They're being pulled apart by the spindle. I'm not drawing the spindle, but just like in mitosis, they're getting pulled apart. And this is anaphase I. We're just separating the homologous chromosomes, and they're going to be going into different cells. Go ahead, take your picture, and then we'll move on to telophase I. Telophase I. Here we go, telophase I. Again, no crossing over. Telophase I, what happens? The sister chromatids kind of, or the chromosomes, they kind of gather, and we have, during telophase I, something called cytokinesis happen. At this point, cytokinesis is separating this one cell into two cells. So moving forward, I'm going to keep this line here the entire time because this is cell number 1. This will be cell number 2. Go ahead, take your picture. We got telophase I. So now after telophase I, these cells are just kind of back in prophase, but instead of prophase I, they go to prophase for the second time, so it's called prophase II. And again, we're doing no crossing over. Here's our cell, prophase II. We have cell number 1 on this side, cell number 2 on that side. They're two entirely separate cells. It's hard to show that on the board here, but they are. That's why this line is still there. Take your picture. Let's look at metaphase II. Still no crossing over. Metaphase II are chromosomes. They kind of move to the center of our cell. And again, this homologous chromosome is not in the same cell as this one, and this one is not in the same cell as this one. Metaphase II, they just kind of line up in the center of the cell like so. Take your picture. Anaphase II. Still no crossing over. Anaphase I, we separated the homologous chromosomes. Now they're in entirely different cells. Can't separate them again, they're already separated. Now we're going to separate the sister chromatids. So we're going to start to separate these ones, and those ones, and those ones, and those ones. They would be moving away from each other, again pulled apart by the spindle, which we're not drawing. But here we got anaphase II. Then we have telophase II. Again, once again, there is no crossing over. Telophase II, these chromosomes kind of gather on opposite sides. We'd have nuclei forming or a nucleus forming. Boom, boom. But again, what happens during telophase? We have cytokinesis. So again, cytokinesis is going to separate these from these, putting these in a different cell than this one. So I'm going to draw cytokinesis right there. I guess I can get rid of cell 1 and cell 2. We got cytokinesis. And then that's going to happen in this cell as well. We got cytokinesis as well. And now what we have are 4 cells. Cell number 1, cell number 2, cell number 3, cell number 4. And when I look at this, I see that each cell has one copy of this chromosome. See, this cell has one copy, this cell has one copy, this cell has one copy, it's just blue. This cell has one copy as well. Also, each cell has one copy of this one. And since there's only one copy of this chromosome, that means cell number 1 is haploid. If there was 2 copies of this chromosome, it'd be diploid. This one, there's only one copy of the chromosome, it is haploid. If it was in a cell with 2 of these, it'd be diploid. Same here, there's only one copy of each of those 2 chromosomes, so it's haploid, and so is our last one, haploid. The process of meiosis, we started with one cell, had 2 copies of each chromosome, so we started with a diploid cell to create 4 haploid